So in our final topic, we take a look at a little bit of nucleic acid and protein synthesis. So let's start with nucleic acids first. And for our nucleic acids, we know that they are made up of your nucleotides. Just in a sense, like proteins are made up of amino acids, your nucleic acids are made up of nucleotides. And let's start with nucleotides first. Nucleotides essentially have three components. You have a nitrogenous base, you have a 5-carbon sugar, and you have a phosphate group. Now, you don't have to memorize the structures of the nitrogenous bases and the 5-carbon sugars. So I'm not going to say draw adenine or draw thymine or draw ribose or such like that. However, with them, there are certain aspects that you need to remember. And you'll see this in some of the problem solving that that we'll do with you in the next 45 minutes or so. But again, you don't have to memorize the structures of the nitrogenous bases. You don't have to memorize the structures of the 5-carbon sugars, but there are certain aspects of them that you should really become familiar with. All right. So let's take a look at our first example. State whether each of the following would be found in RNA, DNA, or both. And remember, in terms of nucleic acids, there are two types of nucleic acids. There's ribonucleic acid and there's deoxyribonucleic acid. Ribonucleic acid, of course, referred to as RNA. Deoxyribonucleic acid, known as DNA. And one of the things that we'll go through as part of our discussions are the similarities and the differences between them. All right. Number one. And let's see if we can write a 1 that looks like a 1. All right, so first one, let's do guanine. So guanine, of course, is what? Guanine is a nitrogenous base. Remember, RNA and DNA have 4 nitrogenous bases. 3 of them are common to both. Is guanine one of those nitrogenous bases that are common to both? And the answer is yes. So guanine would be found in both. So that's number 1. Number 2. Thymine. Now, thymine is also a nitrogenous base, but it's actually one of those nitrogenous bases that's not common to both. So, in which nucleic acid in the nucleotides that are found in them could you find thymine? Would they be found in nucleotides found in RNA or nucleotides found in DNA? Thymine is a potential nitrogenous base that would be found in DNA.
Is there a trick to know this or memorization?
Yeah, just really got to memorize it. You really just have to know that adenine, cytosine, and guanine are common to both. And then thymine is the 4th nitrogenous base in DNA. And, uracil is the 4th nitrogenous base in, RNA. If you want to sort of remember RU, So like RNA uracil, you could try to do it that way, but really that just comes down to memorization.
Okay, thank you.
You're welcome. Number 3. Adenosine monophosphate. So adenosine monophosphate is actually the name of a nucleotide. You saw in the chapter a little bit about naming, where for the name of a nucleotide, you take the name of the nucleoside and you add the ending monophosphate, or how it was taught at one point, 5' monophosphate. Now, one of the tricks to solving this one is, yes, adenine, because adenosine refers to which nitrogenous base. Adenosine refers to adenine. And remember, adenine is one of those bases that's common to both RNA and DNA. But the trick here is that adenosine monophosphate, the nucleoside name adenosine, identifies it as being in what? It identifies it as being in RNA. Because remember, if you think about the nucleoside name, the difference between what you would have in DNA versus RNA is that in DNA, in front of the nucleoside name, you would have the prefix deoxy. When you don't have the prefix deoxy in front of the nucleoside name, then it refers to something in RNA. So if you had deoxyadenosine monophosphate, the answer would be DNA. But since here you only have adenosine monophosphate, that refers to RNA. Not both, not DNA, RNA here for number 3. And let's do one more, number 4. Let's do deoxy, guanosine, monophosphate. Now again, this is another name of a nucleotide. And if we think about the nitrogenous base, guanosine, guanosine refers to which nitrogenous base? Guanosine refers to guanine. But here, if we focus on the nucleoside portion of the nucleotide name, we have the term deoxyguanosine. So in front of the guanosine, we have the prefix deoxy, which is the indicator that this nucleotide would only be found in DNA. Again, if you had guanosine monophosphate, that would be found in RNA. But if you have deoxyguanosine monophosphate, that would be found in DNA. So, in terms of the nucleoside names, and again, you don't have to memorize the nucleoside names. But if I gave you something like adenosine monophosphate and asked you if it's an RNA, DNA, or both, you should be able to tell me RNA. If we give you deoxyguanosine monophosphate and ask you if it's an RNA, DNA, or both, you should be able to tell me DNA. Okay. So keep that in mind. So that's our first example. All right. Second, draw a nucleotide made from the following components. Let's see. Here's our first component. Actually, you know what? Change it up a little bit. Sorry about that. Second component. And then here's our third component. So I can just, all right. So what do we got here? We've got base, we've got sugar, we've got phosphate. So the three components of a nucleotide are, of course, phosphate, sugar, and base. And if we give you the three components of a nucleotide, we need you to be able to put them together in order to give us our nucleotide. So, a couple things. First off, in terms of the nitrogenous bases, you need to know which nitrogens are going to be the ones that are going to be linked up to the sugars. And if you go back into the notes, we circled the hydrogen on the nitrogen for each of the nitrogenous bases. That would be removed when the base becomes connected to the sugar. Again, you don't have to memorize or know what the name of this nitrogenous base is, but if you look at its structure, you need to remember that the nitrogen that's going to become connected to the sugar is this nitrogen right here. And it's going to be this hydrogen here that's going to be removed. So again, when you go back and look at the notes involving the different nitrogenous bases, you need to know which nitrogens are going to become connected to the sugars and which hydrogens are going to be removed in order for that to occur. Now, let's go to the sugar. If we're making a nucleotide, there are two important carbons that we want to remember. And that's carbon 1' and that's carbon 5'. And remember, the carbons in the sugars are numbered with primes to distinguish them from the carbons in the nitrogenous bases. We know that at carbon 1', you're going to have the link to the nitrogenous base. And at carbon 5', you're going to have the link to the phosphate group. Essentially, each of those linkages involve removing water. Because essentially off of carbon 1 prime, I'm going to take off the OH, and I probably should have circled that. So the OH and the H, that's going to be one water that's going to be removed. And then for the phosphoric acid, we're going to take off the OH, and off carbon 5 prime, we're going to take the hydrogen, and let me just see if I can write on 5 prime a little bit better. Sorry. Off of carbon 5 prime, we're going to take off the H. So essentially those, and I should have squared that instead of circling it, of course. Those are going to be the waters that are going to be removed. Okay. So going from left to right, we're going to do phosphate, sugar, base. All right. So we'll start with phosphate first. And the part that's going to be removed is the OH off of the phosphate group. That's going to become, the phosphorus is going to be connected to the oxygen on carbon 5 prime. So the phosphorus becomes connected to the oxygen on carbon 5 prime. To give you what type of functionality? To give you what is known as a phosphate ester. P double bond O, O, C, phosphate ester. Then I'll fill in the rest of the sugar. Give myself a little more. Now, once I get to carbon 1 prime of the sugar, when the OH comes off and the H comes off the nitrogenous base, I'm going to connect carbon 1 prime to the nitrogen, the correct nitrogen on the nitrogenous base. And then I'm going to have the rest of the structure of the nitrogenous base. And the connectivity between the sugar and the nitrogenous base is another example, in a sense, of a glycoside bond. It's different from what we saw for carbohydrates. For carbohydrates, we saw ether type functionalities. But here in general, we still have another example of a glycoside bond. Okay. And this would be the nucleotide. Phosphate, correctly connected to sugar, correctly connected to base. Now, one of the other questions that's usually asked is that, is this nucleotide found in RNA or DNA? Now, there's a couple ways to answer this question. One way would be if you recognize which nitrogenous base we're using here. I'm not telling you which one it is. And again, you don't have to memorize the nitrogenous bases, so you might not know exactly which nitrogenous base it is. So then the question becomes, how do you identify it? And what you have to remember to do this is that the difference between ribose and deoxyribose is in the downwards direction off of carbon 2 prime. For ribose, what goes in the downwards direction off of carbon 2 prime OH for deoxyribose, what goes in the downwards direction of carbon 2 prime? H. So since we have an OH going downwards off of carbon 2 prime here, it indicates that this nucleotide would be found in RNA. The nitrogenous base that I used here was uracil. And remember, uracil is only found in RNA. So if you recognized it as uracil, you would know that this would be found in RNA. But really, for the way I sort of remember it, is just keep in mind the difference between ribose and deoxyribose, which would be at carbon 2 prime. Right? At carbon 2 prime.
So for the test, do we have to draw this out or just be able to pick things off of it?
No, you should be able to draw it out. If I give you the sugar, the nitrogenous base, and the phosphate group, you should be able to put them together to come up with a nucleotide.
Okay, is this going to be a big part of the test or just a small section?
So it would just be like one question on the final exam to do this.
Okay, thank you.
No problem. Yeah, but this will sort of just be one question. And again, I'll talk more about that when I post my announcement later on this evening. All right. Just to also mention, because I think it's important, when you so this is how you put nucleotides together, or how you put a nucleotide together. One nucleotide, sorry. When you link nucleotides together to give a nucleic acid, remember the connectivity that you end up having really connects the 3' carbon of the sugar in one nucleotide to the 5' carbon of the sugar in the next nucleotide. And as we saw in the notes, and I'll just sort of circle it here, the connectivity that you have that links nucleotides together is an example of what is known as a phosphodiester bond. Where do we see phosphodiester bonds previously? We saw phosphodiester bonds previously when we talked about glycerophospholipids and sphingomyelin. So here, for phosphodiester bond, you have P double bond O, OC once, and then you have P double bond O, OC twice. That's your phosphodiester bond. And you have this when you link nucleotides together. You know, we talked about when you're putting a nucleotide together, you know, the phosphate connects to the sugar with a phosphate ester. The sugar connects to the nitrogenous base with a glycoside type bond. But here, when you link nucleotides together, you get a phosphodiester bond. Now, the difference between nucleotides in a nucleic acid is the nitrogenous base that is present. Because think about it. If you are in DNA, for each and every nucleotide, the sugar's got to be the same, deoxyribose. Phosphate is always the same. What's the only variable from one nucleotide to the next? The only variable is the nitrogenous base, because there's 4 different ones. You know, if we're talking about DNA, there's cytosine, guanine, thymine, and adenine. That's the variable. As you go from one nucleotide in a nucleic acid to the other, the variable is the nitrogenous base that's present. So keep that in mind as well. All right. So let's talk a little bit more about DNA. And in our next example, let's write the complementary base sequence for the following DNA segment. So remember, the structure of DNA is essentially a double helix. 2 what are known as polynucleotide strands, in other words, many nucleotides linked together, winding about each other like a spiral staircase, where on the outside of the staircase, you have the sugar and the phosphate group, so they're like the railings of the staircase. And then you have the nitrogenous bases sort of arranged as steps on the inside. And what holds the 2 strands together in the double helix is hydrogen bonding between nitrogenous bases. And one of the key points is that the hydrogen bonding between nitrogenous bases is very specific. We talked about, or you saw in the notes, that adenine only hydrogen bonds with thymine, and cytosine only hydrogen bonds with guanine. And essentially, these 2 combinations, adenine with thymine, cytosine with guanine, are what are known as complementary base pairs. Now, you don't have to identify the hydrogen bonding in each. You know, what you'd want to remember is that for adenine and thymine, you have 2 hydrogen bonds between them. We're not going to ask you to sort of draw it out, but you just want to remember that there's 2 hydrogen bonds between them. And then for cytosine and guanine, there are 3 hydrogen bonds between them. But again, the hydrogen bonding is very specific. Adenine and thymine will only hydrogen bond with each other. Cytosine and guanine will only hydrogen bond with each other. So if you had the following DNA segment, and we'll use the 1-letter abbreviations, ATA, GCCC, TTA, CTGG, got it all. What would be the complementary base sequence? So if you had adenine, so we'll do the original on the top, the complement on the bottom. So if the original has adenine, your complement is going to have thymine. If your original has thymine, your complement is going to have adenine. Then we come across adenine again, which would give us thymine. We have guanine, which gives us cytosine. Then we have 3 consecutive cytosines, which would give us guanine, guanine, guanine. Then we have 2 thymines, which would give us 2 more adenines. Then we have adenine, which would give us thymine, cytosine, which would give us guanine, thymine, which would give us again adenine, and then guanine and guanine, which would give us cytosine and cytosine. Okay, so one of the things that you do have to remember is you do have to remember these complementary base pairs as you go from one DNA segment, or as you have these complementary, if you have in the DNA double helix, you have 2 complementary DNA strands, which are held together by hydrogen bonding between complementary bases. So keep that in mind. Your DNA double helix, 2 complementary DNA strands winding about each other like a spiral staircase, held together by hydrogen bonding between complementary bases. The other thing I wanted to mention in terms of DNA, I just wanted to mention a little bit about DNA replication. So keep in mind with DNA replication is that the 2 strands in the original, or what is known as the parent DNA molecule, separate. Enzymes are going to break the hydrogen bonds that exist between the complementary bases. But here's sort of the key idea. The 2 separated strands don't make copies of themselves. They make copies of their complementary strand. But since the 2 strands are complements in the original or parent DNA molecule, since the 2 strands are complements of each other, even though they're not making copies of themselves, even though they're making copies of their complementary strand, both original strands end up being copied. The 2 original strands just don't make copies of themselves. The 2 original strands make copies of their complementary strand. And what you get is you get 2 new DNA molecules identical to each other, and the parent DNA molecule called daughter DNA. Where each has one original strand and one new strand. So every time a DNA molecule gets replicated, you don't get one molecule, you actually get two DNA molecules. These two new molecules are what are called daughter DNA. The two daughter DNA are not only identical to each other, but they're also identical to the original or the parent DNA. And the daughter DNA, just like the parent, has two strands, one original and one new strand. The copy of the complement, the new strand, is the copy of the complement it made. So the original strand makes a new complement, and then they become hydrogen bonded to each other. To give it, it's not as simple as that, of course, in a sense, but that's essentially what occurs in terms of DNA replication. But one of the mistakes students commonly make is they think that the original strands make copies of themselves. No, the original strands make copies of their complementary strands. All right, good. All right, so as promised, we're going to do a couple of examples dealing with RNA and the genetic code. So first off, let's just talk a little bit about the different types of RNA that you come across. And there are essentially three main types of RNA. Let's go to color. All right, you have your ribosomal RNA, which is commonly known as rRNA. You have your messenger RNA, which is what is commonly known as mRNA. And then you have your transfer RNA, which is commonly known as tRNA. And essentially, all three of these types of RNA are essentially involved in protein synthesis. Now, and just like in the notes, in a sense, we're not going to go through every single aspect of protein synthesis. We'll talk a little bit about transcription and essentially translation, the two sort of main steps associated with it, and we'll provide some details, but we're not going to go through the entire process. When I think I spelled ribosomal RNA wrong, so let's just fix that. So I had too many S's. Ribosomal RNA. So from my perspective, in terms of the three different types of RNA, the two main players in protein synthesis are really the messenger RNA and the transfer RNA. And as we'll talk about in a few minutes, one of the things to understand about protein synthesis is the idea that the messenger RNA calls for the amino acids to be brought to the ribosomes for protein synthesis, but the transfer RNA actually brings it. And we'll talk a little bit about the relationship between messenger RNA and transfer RNA as well. So essentially, to talk a little bit more about the three different types of RNA, let's take a look at an example question. All right. So in this question here, what we want to do is we want to match the following statements with ribosomal RNA messenger RNA or transfer RNA. All right. So letter A carries genetic information from the nucleus to the ribosomes essentially for protein synthesis. So essentially, which one of these three types of RNA does this statement refer to? This statement refers to messenger RNA or mRNA. So essentially, what you have is you have a DNA segment that unwinds. And a portion of that unwound section of essentially one nucleotide strand is used to make a messenger, RNA. All right. And again, in going back to DNA for a second, each DNA molecule is a double helix that has two nucleotide strands winding about each other like a spiral staircase. As we talked about previously, the force of attraction that you have between the complementary bases on the strands is hydrogen bonding. That force of attraction holds the strands together. Well, essentially, an enzyme is going to break the hydrogen bonding in a portion of that DNA double helix. And as we'll sort of get into a little bit, there's going to be sort of an identification point of where sort of messenger RNA, where the messenger RNA should start to be made from in a certain sense. Okay. So you have a DNA segment that unwinds by breaking the hydrogen bonds between complementary bases, not the entire DNA double helix, but a segment of it. And then a portion of that unwound section of one nucleotide strand is used to make a messenger RNA. Now, what's the relationship between DNA and messenger RNA? DNA and messenger RNA are complements of each other. And this is really very similar to the idea of the complementary base pairs that you have in the DNA double helix. Essentially, if you consider cytosine, guanine, thymine, and adenine, the ideas of complementary base pairs that we see in the DNA double helix hold true, except for one big difference. So if your DNA has cytosine, your messenger RNA is going to contain guanine. If your DNA contains guanine, your messenger RNA is going to contain cytosine. If your DNA contains thymine, your messenger RNA is going to contain adenine. But if your DNA contains adenine, your messenger RNA is going to contain uracil. This is the difference in terms of comparing complementary bases in the DNA double helix, because RNA doesn't have thymine. Instead, RNA has uracil. So this is the one difference. And we'll apply this to a question that we'll do in a little bit. All right. So that's the first statement, letter A. There's some little bit of other information about it. All right. Letter B. Brings amino acids to the ribosomes for protein synthesis. So as we mentioned at the beginning of the video, your messenger RNA calls for the amino acids to be brought to the ribosomes for protein synthesis. But what actually brings the amino acids to the ribosomes for protein synthesis is your transfer RNA. So your transfer RNA would be the answer here. Now, what's the relationship between messenger RNA and transfer RNA? Well, messenger RNA and transfer RNA are essentially complements of each other. So just to sort of expand a little bit, on the table above, we can fill in for transfer RNA. If your messenger RNA contains guanine, your transfer RNA is going to contain cytosine. If your messenger RNA contains cytosine, your transfer RNA is going to contain guanine. If your messenger RNA contains adenine, your transfer RNA is going to contain uracil, right? No, thymine. And if your messenger RNA contains uracil, your transfer RNA is going to contain adenine, because messenger RNA and transfer RNA are complements of one another. Letter C. Combines with proteins to form ribosomes. Combines with proteins to form ribosomes. This would be your ribosomal RNA, or your rRNA. Combines with proteins to form ribosomes. This is your ribosomal RNA. Letter D. Acts as a template for protein synthesis. And essentially, the template what essentially we look at to figure out what amino acids are going to be brought to the ribosomes is going to be your messenger RNA. And then finally, letter E makes up the highest percentage of RNA in the cell. And essentially the most abundant type of RNA that we have is the ribosomal RNA. All right, so hopefully just by looking at these questions and with a little bit of extra in-depth information, we now have a pretty good idea of the 3 different types of RNA that exists. All right. So as I mentioned a little bit earlier, the 2 key steps in protein synthesis are first transcription. And essentially in transcription, DNA is made, or I should say, let's say it this way. Messenger RNA is made from a section of DNA. Remember, one of the key differences between DNA and RNA is that RNA tend to be much smaller than DNA. And one of the ways I always say that you can rationalize this is based on the fact that messenger RNA is made from a section of DNA. So it's one way to rationalize it. The other key difference, besides the other ones that we've talked about in the previous video we did, the the previous Zoom session is that RNA molecules are single-stranded versus DNA molecules, which are double-stranded. That's the final key difference. So in transcription, messenger RNA is made from a section of DNA. And then essentially you have your translation, where the messenger RNA calls for amino acids and we use what is known as the genetic code to interpret the sequence of bases in sets of 3 called codons. And then your transfer RNA brings the amino acids to the ribosomes. So again, this is a key point. The messenger RNA calls for the amino acids, the transfer RNA brings it. But how does the messenger RNA call for the amino acids? Essentially, the bases that are found in the messenger RNA, because remember, they're made up, just like DNA, they're made up of nucleotides. So you're still going to have phosphate, sugar, and base. Just like we mentioned how if you think about DNA, the only difference between nucleotides would be in that DNA would be the nitrogenous base present. Same thing for RNA. The only variable that you have in going from one nucleotide to the next in an RNA molecule would be the nitrogenous base. In what is known as the genetic code, bases are not read individually. Bases are not read in pairs. They're read in sets of 3. And each set of 3 is what is known as a codon. Now, in the notes and in your textbook, you have the genetic code. You don't have to memorize it. For the final exam, I will give you a copy of the genetic code as well to solve problems. You don't have to memorize it. Couple points. First off, if you look at the genetic code, there are a total of 64 codons. There's only a total of 20 amino acids. So as the genetic code easily illustrates, since basically we have triple the number of codons than we have amino acids, almost all amino acids have multiple codons that code for them. Then we also have the start codon. Our start codon is the signal for protein synthesis to begin. And there's only one of them, and it is AUG. And what you notice is that it codes for methionine. So essentially, as the protein is being made, for every instance, the first amino acid present is going to be methionine. But by the time all is said and done, that initial methionine that's found is usually removed. But as essentially the protein is being made, the first amino acid that you're going to come across is going to be methionine. But again, before all is said and done, then basically that initial methionine at some point is going to be removed. And then on the other hand, you have your stop codons, which signal protein synthesis to end, because they do not code for any amino acid. So no amino acid is going to be brought by the transfer RNA. And that's the signal for protein synthesis to come to an end. Which are the stop codons? you have, and that looks too much like a V. So you have UAA, you have UAG, and you have UGA. And you don't have to memorize these. You know, it's clearly illustrated on the genetic code, which ones are the stop codons and such like that. All right. So you have transcription, where you have the messenger RNA being made from a section of DNA. And then essentially you have translation, where the messenger RNA will call for the amino acid to be brought to the ribosomes, and the transfer RNA will bring the amino acids to the ribosomes to make the protein. Again, we're going to keep it very generalized. You know, we're not going to get into all the specifics in terms of essentially initiation, chain elongation, things of that matter. We're going to just keep it to a very broad level. So let's take a look at an example. Give the corresponding messenger RNA, transfer RNA, and amino acid sequences for the following segment of DNA. Erase the whole thing. All right. So actually, let's do that. Let's do that example again. And let's do it the right way. So DNA is going to contain A, T, G. Then we've got C, C, A. Then we've got C, C, T. Then we've got A, A, A. And then we've got A, T, T. All right, that's better. Hopefully you caught that mistake, but if not, you know, there it is. And I'll see if I can actually edit the video to take out that problem, but we'll see what we can do. All right, so we got messenger RNA, then we've got transfer RNA, and then we've got the amino acids. All right, so we'll have, remember, DNA, messenger RNA, complements of each other. So we've got U, A, C, G, G, U, G, G, A, U, 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 A, A. All right, now let's go from the messenger RNA to the transfer RNA. So uracil would give us adenine, adenine would give us uracil, cytosine would give us guanine. Guanine would give us cytosine, uracil would give us adenine. Remember, messenger RNA and transfer RNA are complements of each other. 2 guanines would give us 2 more cytosines. The adenine would give us uracil. The 3 uracils are going to give us 3 adenines. And then finally, the UAA will give us AUU. All right. The amino acids. And this is really, when I graded the homeworks a little bit, I've seen the mistakes. Because what some students are doing is they're using the transfer RNA. No. When we use the genetic code, remember, the genetic code is based on the messenger RNA sequence. So we're not going to use the transfer RNA sequence. We're going to use the messenger RNA sequence. All right. So I'm looking at the genetic code on another screen. I find UAC. And when I come across UAC, and the 3-letter abbreviations are perfectly fine. I have no problem with that. So UAC gives us tyrosine as our first amino acid present. GGU would give us an amino acid of, sorry, not GLU would give us GLY. And let's put the pen back. Okay. But it's still there. Let's get the pen back. All right. So it'll be GLY. I don't want subtitles either, but let's see if we can figure out. There we go. Knew it had to be something I hit. GLY, which of course is glycine. GGA. GGA would also give us glycine, and I still didn't get the pen back. There we go. Would give us glycine. So we would have two glycines back to back here. UUU would give us phenylalanine, PHE. And then UAA would actually be a stop codon. So we would have a signal to come to an end. All right. So tyrosine, glycine, glycine, phenylalanine, and then stop. And again, the key in terms of determining the amino acid sequence was that I used the messenger RNA. Right? So don't use the transfer RNA, use the messenger RNA. And again, you'll have a copy of the genetic code, so you'll be able to use that to figure out the amino acids, and the 3-letter abbreviations are perfectly fine. All right. So I hope these couple of problems help, and I wish everybody